Hi everyone, I'm Dua Kasim, and today I am sharing a really exciting study from Cell called Mapping the Human Gut Microbiome, insights from 168,000 people around the world. What pulled me into this study is how it flips the usual script. Most microbiome research focuses on people from industrialized countries, which leaves out a huge portion of the world and a lot of microbial diversity. That creates a very incomplete picture of what's normal or healthy. The authors wanted to fix that. They pulled together data from over 168,000 people in 86 countries, reanalyzed it all with the same method, and asked, what's really shaping our gut microbes? Is it geography, diet, or industrialization? And can we use that info to better understand health in a more inclusive global way? It's not just about the numbers, it's about the vision. This study built something like the Human Genome Project, but for the global gut. So, over the next few slides, I will walk you through the background, what they did, and what they found, and why this global lens really matters for microbiome science and for public health. Before diving into the data, I want to pause and talk about why this study actually matters. We all hear that the gut microbiome is linked to digestion, inflammation, even mental health. And yeah, that's true. But we don't always stop to ask, where is this information coming from? Most of it comes from people in industrialized countries, the US, Europe, parts of East Asia. That means the global picture we have built so far is incomplete. It's kind of like trying to understand all of humanity by only studying a few wealthy cities. This study set out to change that. The researchers pulled microbiome data from over 168,000 people in 86 countries, and instead of just combining the results, they ran every sample through the exact same analysis pipeline. That's huge. It means you can finally compare things fairly across regions and lifestyles, no more apples to oranges. They ask some big questions. What does the global gut microbiome actually look like? How do geography, lifestyle, and industrialization shape what lives inside us? And can we use that knowledge to improve health everywhere, not just in the West? That's the motivation behind this work and why it matters beyond just the science. And yes, they found real meaningful differences. Microbes like Brevotella were more common in traditional, non industrialized populations, while other species showed up more in urban, westernized environments. One of the most exciting parts, they trained a machine learning model that could actually predict where someone lived based on their gut microbes, and it worked surprisingly well. So this was not just about collecting more data, it's about building fairer, more global microbiome science. It gives us tools to understand gut health in ways that reflect all kinds of lives, not just the ones we have always studied. To really understand the importance of this study, we need to zoom out and think about where microbiome research has been going. The gut microbiome is not just about digestion anymore. It's connected to immune function, inflammation, and even how we respond to different treatments. It's one of the most exciting and complex areas of biology right now. In 2021, Wastek and colleagues showed that changing your diet, things like eating more fiber or fermented foods, could shift your gut microbiome and actually reduce inflammation. That's huge because it suggests we can support the immune system through food. Then we have work like Jibril and colleagues 2022 who explored the clinical potential of targeting the microbiome for disease treatment. But they also raised an important red flag. Most of the microbiome data we rely on still comes from industrialized countries. So global health strategies are being built from a limited and possibly biased view. And even when data is collected from around the world, there is another issue. Different studies use different tools and protocols. Comparing them is like comparing recipes written in different languages. That's what makes this new study by Abdul and colleagues 2025 so powerful. They did not just collect more samples, they reprocessed 168,000 microbiome data sets from 86 countries using the same standardized pipeline. For the first time, we get to see global microbial patterns without all the technical noise. It's not just about a big data set, it's a global reset, a way to see the microbiome through a cleaner, fairer, more unified lens. Imagine trying to understand the human culture, but you only interview people from two cities. That's kind of what microbiome science was like for years. Most of our data came from industrialized countries, mostly the US and Europe, so the rest of the world was barely represented. But we already know that diet, environment, and lifestyle have huge effects on the gut microbiome. That means the view we had was limited and likely biased. This study asked what happens if we zoom out? What if we gather tens of thousands of samples, not just from cities but from rural regions, different continents, different ways of life, and run them all through one consistent, standardized pipeline? The goal was not just to catalog microbes, the authors asked deeper questions. Can we actually find patterns shaped by geography, lifestyle, and industrialization? Can we reduce the technical noise that makes global comparisons so difficult? And can machine learning tell us where someone is from just based on their gut bacteria? These are not just science questions, they are questions about identity, equity, and how we understand human health from a truly global perspective. This project didn't start in a lab. It started with a mountain of raw sequencing data from over 120 different studies around the world. That's gut microbiome samples, each collected using different tools, protocols, and standards. In other words, total chaos. The researchers wanted to make meaningful comparisons, so they reprocessed all the samples using the same pipeline, a tool called Biome Scout, and then used MetaFLAN-4 to figure out which microbes were present. Instead of sequencing everything, MetaFLAN scans for specific microgenes, kind of like microbial barcodes, to identify species quickly and precisely. But reprocessing the sequences was only part of it. They also had to organize the metadata information about each person's location, age, and whether they lived in rural or urban area. They grouped all of this into broader categories like geography, lifestyle, and industrialization level, which made it easier to compare samples fairly. Then came the analysis. They used something called Hsien distance, a method built for compositional data, like microbiomes where the parts always add up to a whole. They applied Permanova to check if differences between groups were actually statistically meaningful. And finally, they used Light GBM, a machine learning model, to see if it could predict someone's background or lifestyle just from the bacteria in their gut. And it worked. They also use tools you might recognize from our own microbiome course. First up, Shannon diversity. It measures how rich and evenly spread the microbes are in a sample. The authors used it to compare diversity within each sample, and guess where some of the highest values showed up? Sub-Saharan Africa. Then there is multidimensional scaling or MDS. If you have seen ordination plots before, this works similarly. It helps visualize how similar or different microbial communities are. Here they used it to compare microbiomes across different global regions. So this slide really shows how the authors didn't just clean and reprogress a huge data set. They used both classic microbiome tools and modern machine learning to build something new, a global map of who lives in our gut. So far we have looked at biology, but this first figure is a bit of reality check. It shows us that how we study microbes can shape the results just as much as where the samples come from. Most of the samples in this dataset were from amplicon sequencing, not a whole genome sequencing. And if you look at panels A and B, you will see that WGS was mostly used in Europe and North America. Other regions, mostly amplicon. Panel C makes this clear. We are not just comparing global regions, we are comparing different technologies. Over time, amplicon studies exploded in number. That's shown in panel D. But that growth didn't always mean better consistency. In fact, panel E shows that most of those studies focused on just one region of the 16S gene, usually V4. That matters because V4 doesn't detect all microbes equally. Panel F shows another imbalance. A few huge projects contributed most of the data, while most studies were pretty small. What I found most surprising is panel G. You would think geography would explain the most variation in diversity, but actually technical details like bead beating had an even bigger effect. And panel 8 shows the consequences. Your choice of primer can shift what microbes you detect, making two identical samples look like they came from different parts of the world. So, the big takeaway before we interpret microbiome data, we have to clean up the technical noise. That's why using a standardized pipeline, like in this study, is such a game changer. It helps ensure global comparisons are actually fair. Now let's take a closer look at how geography shapes the gut microbiome. In panel A, you can see where all the samples came from, a pretty solid global spread, though not totally even. Panel B shows the sample counts by region. Some areas like Europe and North America are more heavily represented, which is good to keep in mind when we look at trends. Panel C is where things get interesting. It shows Shannon diversity across regions and the highest diversity. It's not in the West, it's in places like Sub Saharan Africa and Southeast Asia. That suggests these communities may carry more diverse microbial ecosystems, maybe because of differences in diet or environment. Panel D is important because it rules out a common concern, sequencing depth. Since depth was pretty consistent across regions, we know the diversity differences are real, not technical. Panel E is multi-dimensional scaling plot. And if you have used these before, you know what we are seeing. Clear regional clustering, it's like each place has its own microbial accent. And panel F reinforces that idea. The density plots show nice separation between regional microbiomes, especially on MDS1 and MDS2. So, what's the takeaway? Where you live matters. Geography doesn't just shape your environment, it shapes your microbiome too. Even after accounting for technical stuff, people from different parts of the world still have distinct microbial fingerprints, likely tied to local diets, lifestyles, and more. Alright, let's zoom in on something super interesting. Not just who is in the microbiome, but how those communities differ depending on where you are in the world. Panel A shows a familiar pattern. Regions like Eastern, Southeast Asia, and Europe, North America have lots of unique microbes. But that's probably because they have been studied more. But then you hit panel B, and it's kind of wild. In sub Saharan Africa, there are way more unclassified microbes. These bugs are there, but we don't know what they are. That tells us we are still missing huge parts of the microbial picture in some parts of the world. Panel C shifts the view from uniqueness to composition. You will see the industrialized places lean heavily toward Bacteroidota, while other regions have more balance and more dominance from less familiar phyla like Desulfobacteriota. Panel D lines up the microbial profile side by side, and the shapes are all different. It's not just which microbes are there, but how much of each. That speaks to structure, not just presence. Then we get to panels E and F. These are microbial co-occurrence networks. It's like mapping out who's friends with who. In Europe and North America, you see one kind of a friend group. But in other parts of the world, the social circles look totally different. Microbes that cluster together in one region don't necessarily do that somewhere else. So, what's the takeaway? It's not just about finding new microbes, it's also about understanding how they work together. Each region doesn't just have different microbes, they have different microbial ecologies. And if we want a truly global understanding of gut health, we have got to include everyone in this story. Alright, now let's talk about lifestyle. This figure asks a simple but powerful question. Does the way we live, rural compared to urban, industrialized compared to traditional, leave a mark on our gut? And the answer absolutely. Panel A kicks it off. People in rural communities had higher microbial diversity. More species, more balance. That's a big deal because diversity often means resilience. Then panel B zooms out. It shows that the less industrialized your environment is, the more microbial variety you tend to have. In other words, modernization might be shrinking our microbial ecosystems. Panel C is fun. It's from the machine learning model. Turns out industrialization was one of the top predictors of microbiome patterns, right up there with geography and age. Panel D is like microbial vibe check. It shows how people cluster based on lifestyle, and it's pretty clear. Folks living in high versus low industrialized areas have very different gut profiles. And panel E and F, they show specific microbes linked to lifestyle. Traditional communities learned toward Prevotilla, while urban ones had more bacteroides, like different microbial accents depending on how you live. So, the big idea, it's not just about where you live, it's how you live. Our microbes pick up signals from our food, our environment, and our daily routines. And as more people move towards urban life, we might be losing some microbial richness along the way. So, this is the slide where everything really comes together. First, the big picture. This study showed that the gut microbiome isn't random. It's shaped by where we live, what we eat, how we live, geography, diet, lifestyle, and how industrialized our environment is all leave a microbial fingerprint inside us. Why does that matter? Because if we are comparing microbiomes across the world, we cannot do it with mismatched tools and pipelines. Without standardization, we are not comparing biology, we are comparing lab artifacts. This paper makes that really clear. Global comparisons only work if the methods are fair. And what is the real takeaway? This is not just about cataloging bacteria, it's about building a framework, one that lets us find patterns that truly matter for health, no matter where someone's from. It's science that includes more people, more regions, and more stories, not just the easiest ones to study. And I want to close with a reflection that's stuck with me throughout this project. Who we study shapes what we find. If we want microbiome science to truly serve global health, then it's time we include the whole world in the picture. Alright, let's pause here and ask, what's actually new? What did this paper teach us that we didn't already know? First, Abdul and colleagues 2025 really shook things up. They showed that gut microbiome diversity is way broader than we thought, especially in regions that have been overlooked. Basically, we have been missing a lot just because we were not looking in the right places. Then you have got Shu and Fellows 2020 who reminded us that it's not just where we live, our own gene influence our microbiome too, and those gene microbe links, they vary from person to person and group to group. Wang and colleagues 2023 added the clinical lens. They found that even when people have the same disease like atopic dermatitis, their microbiome looks totally different depending on where they are from. That's a big deal for personalized medicine. And finally, Safarchi and Fellows 2025 flipped the script a bit. They showed that microbial resilience and dysbiosis are not universal. Something that looks harmful in one context might be totally fine or even helpful in another. So, what's the big takeaway here? Microbiome science is not fully global yet, but it could be. If we start including the rest of the world in our research, we will finally get a fuller, fairer picture. Let's zoom out and think about why all of this actually matters. First, global bias. So many discoveries about the gut microbiome come from just a few places, mainly Europe and North America. But Abdul and colleagues showed us that what we find depends on where we look. If we are only studying a narrow slice of the world, we are missing huge parts of the microbial picture. Second, cultural context really matters. Think about it. If someone develops a probiotic or recommends a diet based on Western data, will it work the same way for someone in rural Asia or sub Saharan Africa? Maybe not. Microbiomes are shaped by what we eat, how we live, even our environment. So, health strategies need to fit the people they are meant for. Third, this study pushed us toward equity by harmonizing data from 86 countries. Abdul and colleagues created a model where everyone's data gets treated fairly. That's how we move from Western centered microbiome science to a truly global one. And finally, standardization really matters. If every study uses different tools, we cannot compare anything. But once you use the same method across the board, the noise disappears and real global patterns start to emerge. So, the broader implication is this: microbiome science is no longer just about bugs, it's about fairness, representation, and designing research that includes the entire world. So, after everything we have learned in this study, where are the gaps and what should come next? First, let's talk about the big unknown, the relationship between genetics, diet, and the microbiome. We still don't fully understand how these pieces interact to shape our health. It's a complicated triangle and it's far from solved. Second, many microbes are still in the shadows, especially in undersampled populations. We have whole branches of the microbial tree that remain unclassified. It's like trying to finish a puzzle when half the pieces are still in the box. Third, even our tools are biased. Master references databases were built using data from industrialized regions. That means when we analyze samples from elsewhere, we might be missing the full picture. We need region specific models to make fair, accurate comparisons. And finally, we cannot assume one size fits all solutions will work everywhere. We need clinical research that tests probiotics, dietary interventions, and microbiota targeted treatments across global populations. Otherwise, we risk creating solutions that only work for a small subset of people. In short, this study opened the door. Now it's up to future research to walk through it. Before I wrap up, I want to leave you with something more lasting than just data. If we truly want microbiome science to serve everyone, we cannot stop at collecting diverse samples and calling it progress. We have to rethink the entire research process, from the questions we ask to the people we engage to the tools we trust. It's about more than inclusion, it's about equity, representation, and shared ownership of the science we are building. So, where do we go from here? This is a call to action for all of us, whether you are a scientist, a student, a policymaker, or a member of the community. Your voice matters. If you want a future where microbiome science is truly global, just and human centered, it will take all of us asking better questions, building better systems, and listening more deeply. Let's shape that future. One study, one step, and one partnership at a time. And with that in mind, I want to share some questions, not just from the article, but ones I hope we carry forward as a community. All right, I will leave you with this. If we want microbiome science to really make a difference, we cannot just aim for big data. We have to aim for big inclusion. This quote at the side of the slide says it perfectly. We cannot stop at collecting diverse samples. We have to build a science that actually reflects the full diversity of people around the world, their diets, their environments, their cultures. So, where do we go from here? We go forward together, whether we are in the lab, in the classroom, or in a community. Every one of us has a role to play in making this science more global, more fair, and more human. And as the quote reminds us, the future of microbiome science is ours to shape. So let's make it count. So here are some questions I'd love for us to think about together. They touch on both technical aspects like how sequencing methods affect results and also the bigger picture like equity and inclusion in microbiome research. One question that really stands out to me is this: can machine learning really capture human complexity without reinforcing existing biases? As we move toward more AI driven tools, this becomes a huge issue. If our training data is already skewed towards certain regions or lifestyles, can our predictions ever be truly fair? I'd love to hear your thoughts, not just on that, but on any of these questions. What excites you most? What concerns you? Let's discuss. And here are the references that supported this presentation. I didn't just rely on the main article. I also pulled from recent studies that explore microbiome diversity, host genetics, data standardization, and the ethical dimensions of global research. They help me connect this work to the bigger picture. Thank you so much for listening. I really appreciate your time and I'm looking forward to your thoughts, questions, or even challenges. Let's have a great discussion.